nema se. Ajde, sačekat ćemo još 2-3 minute da svi okupe, pa ćemo onda nastaviti. Vjerojatno niko više neće doći, tako da možemo početi. Malo kakvih pitanja možda, prije nešto počnemo oko prezentacija, ocjena i tako to, ili sve ok? Dobro. So basically, I don't know if you remember, but as I think at this point several times I somehow tried to elucidate the point that this course by itself is supposed to give you a broad understanding of the possibilities which you can indulge in if you are really interested in learning this stuff, right? The purpose by itself of you learning, in particular, those things by heart is besides the scope of this course. But if there is one message and one thing which I would really like you to take home and understand a little bit more and into more detail, and that's I think why I'm so insistent in talking about those, is data formats. And data formats are very crucial in this field of bioinformatics, but especially in genomics. In general, also proteomics and anything which generates a lot of data, which is basically in this well-known text format, which can be cross-analyzed and merged and analyzed with several different types of data and conditioned to with different data sets to make those things interchangeably analyzable, right? So there are different types of data to save. Each has standard formats. Always use standard formats and/or extend standard formats. Never save data in non-standard formats, not reusable. So what does it mean? So basically, we know that we have all those formats which we mentioned: BIM, BAM, FAM, DOS, BED, SAM, BAM, I don't know, a lot of them, right? And kind of roughly speaking, if you would type the extension txt on it, wouldn't change much of the file. In general, it wouldn't change anything. You could technically still use it. But the point being that if you remember that when you see those text editors, which we kind of specially use, and especially when you upload your data into the interface or open your data with the interface, small changes like saving a BAM file in another format, even though the file doesn't change its properties, doesn't seem like much, but when you upload it into a tool, it mostly doesn't work. So always use the option of saving the file in the suggested output format, right? And the formatting will be standardized so that you can technically analyze it on any machine at any time. Meaning that even if you would export it in a non-standard format by hand, let's say, you would still be able to analyze it on your computer because of some spillover memory from the files you used and tools you used. But basically, if you would try to send it somewhere else or get a different opinion, there is a high possibility of it not working. And there you have a link with a complete format list of formats where you can see what format does and why what format is used. But roughly speaking, in genomics, you kind of have your BAM files and FASTQ and VCF, VCF being kind of the final variant calling file. But we'll talk a little bit more into details about those things during the presentation. So any sequence of nucleotides is saved. This is saved, not saved, in FASTA format of FASTQ, where you have a sequence head with the comments which mentioned in the nucleotide line 1, 2, 3, and so on. Lowercase letters mean they have been masked. So this is how you have a FASTQ format, or FASTA format in this case, where you can see, just a second, how the sequence looks. And with this is mRNA, for example. And then this is something, and this is good for you to have this command is also written here, because we use this in some tools. This is our indexing tool to get a phi index. You can use some tools phi index in your file in the region you want to phi index, and also use bed tools to get this kind of command line for the same thing. Or this is basically the output format which you want to ascribe with bed tools. So this is your input file, and then this line changes the output file you want. It's basically a quick and basic conversion of one file to the other. But keep in mind, even though you can convert your input file to a VCF file, you didn't do all the quality control steps in between. So just having this data into this format without the intermediate quality control doesn't help you much, right? So ideally, you want your FASTQ file, all the steps which we did, and then some, and then get the VCF file, even though technically you could be able to just switch formats, but then again, the data inside of the done VCF file wouldn't reflect the reality of the stuff you would like to analyze. So each read should have a unique name. A read has the sequence and the quality of each nucleotide sequence. So this is basically just, again, telling you the length and some data of the sequence which we have a look into, or which we sequenced. Here you can see it in this case, for example, a lot of missing data in our file. This is our FASTQ file in this case. So we have the sequence of 125 characters, and here we have a lot of missing in the supplementary. So here's the FASTQ1 and FASTQ2 file. If you remember, when we need to merge those, this should be the file from one direction, and this is the file from the other direction. And that's how they work kind of hand in hand to get new to the sum file with the commands we used. So the alignments, once you align you to the reference genome, they are kept in the sum format. Use some tools to view, sort, merge, index, get statistics, and alignment files, which is sum basically. Sum is sorted and compressed into BUM or CRUM. For algeflies, one line per alignment is what the file does and what the format is. The header lines start with an ed. So you have the ed, which is the HD header definition, the HCSQ, a sequence in the reference file you used, followed by how long it was and its comments from the reference file. So the RG, which had problems, if you remember, with the line, read groups you assign while mapping the reads. We did a wrong assignment there. And we have the PG, which is programs used to obtain this BUM. And here's some brief description of everything you encounter in such a file. So with the queue name and the string, this is the types and the ranges which can be used in the queue name. Is a query template name. Then we have a flag, which is an input type, and this is the range of characters you could expect in that line, and it's a byte-wise flag. So we are our name, which is a string of characters, and this is the string here, and this is the reference name, reference sequence name. Then a position, which is integer, right? One based leftmost mapping position. Map q is also an integer. This is a mapping quality, and then we have a cigar, which is also a string. This which is called the cigar string. You can delve deeper into this once if you're interested and see what the cigar string is. And then we have all those zeros. So basically, you differentiate between integer and string in order then to annotate one of those fields. And once you know if it's an integer and a string, you can vaguely associate what kind of data goes inside and what kind of data is preferably written inside of those lines, and you can spot easier mistakes. So the flag is this summation of the following binary attributes. So you have that's the binary attribute. I think we should go really through the detail, but this is a very important one, which is optical or PCR duplicates, and the sequencings which we exclude with this Picard program is a very important step. So that it gets more condensed and easier to use. Yeah, you can read it through in the presentation. I won't bother you going through all of this, and this is the SAM format. And I think at this point we open it several times, so you know already what's in those formats. But in order to open those big files, you need those particular file readers and openers. It's not possible to open them in normal text editor, and also you can't really open it on a slower machine, because at the time it needs to open the file, we just crash the computer in the meantime. So use sum to view to see the content of a sum, bum, or cram file. Always sort, compress, and index them. Use sum tools view, f include, and f exclude to filter by flags, and then you can to see your flags. Let me just open it in parallel, maybe. So here you see all the flags. You just type the sum tool option, and you can see all the flags which are present there. Yes. And then given those options you've seen now, when you type sum tools, you can use those options in order to further compress or look into your file. And extract sequences from BAM file, you can use it by sum tools view, then your BAM file, and then the region you want to extract, chromosome, this and this. But you can also use bed tools with the command intersects in the options, abam, input. I rarely use bed tools. Sum tools is kind of the preferred style, which I use for doing this analysis. And you can go back and convert your BAM file to FASTQ. So assume you have a line that is set in BAM format and want to extract the reads to process the raw data and trim some reads or subsample the length. You can use it in BAD tools with this following command, and you can play a little bit around with this if you want. Once you have this file, it's quite easy to just copy the commands. And this is the command line in sum tools and BAD tools for doing so. This is also standard format for keeping tables. So enter field separated by character on each line. So we have comma separated vectors and tab separated vectors, TSV or CSV files. Some interpreters take any spacer tab as a separator, such as AWK or cut. So when you use those small programming languages, AWK or cut or grep, it's actually quite important to know which of those formats you use: CSV, TSV, TXT, tab, and so on, because commands will differ. Because the programming language will assume certain space in between characters or a string or integers, and if it's not present, it will cut it in real places, and you'll get very odd numbering, and everything will be shifted up and down left to right. So you need to always think about, that's what we're talking about, standard data export. And what format did you export? If you didn't have the right format and those spacing is skewed, you will experience a lot of troubles in trying to align data later on. And then you can, standard format for keeping tables, you can use cut to get a specific field, and AWK or GWK to extract fields and manipulate them. You can use sort. This is all in the Linux interface, right? Sort K to sort the file by a certain field. Use join to join two files by a certain field. And you can all Google list and AWK grep, those small programming languages which are in this interface. Let me see if you can just call it up like this. Yes, you can. So this is AWK. And you can see we have also grep. Yeah, can't use it without a file. We have Perl. Perl is quite big. Shouldn't have done this. But you see what I mean. So the AWK example, for example, we have get an average insert fragment size from a BAM file with the first megabyte of alignment. So with this is output file separator. This is obviously comma delimited, right? This is if you would have tab, you press tab, or space, you press space or down space. And this is just a small command line with your input BAM file using AWK to get your files out or insert a fragment in that given position. You can just copy from the presentation this stuff and try to play around a little bit with it. So genomic variants, so save them in VCF format, VCF variant calling format. The current version is initially 4.4. It's a little bit more advanced. And they are tab separated. And this is how your VCF file looks. So the mandatory header line information about the field column, starting with info, and extra filtering metadata tools. So meta information is basically what everything would happen in the meantime. So allele frequency, total depth of sequencing, answers to allele, TP SNP membership if it's annotated or not, HapMap. Also it tells you some operation less than 50% sample data, and then the genotype, how they look. Here's the headers and here's the body of the data. It's chromosome position, ID, reference, and this is the quality of the sequencing. Then if it has passed our quality control, and this is your genotype as GT, which is quality score and depth and something I forgot. And this is how you can compare a normal tissue, for example, in tumor with the same patient. And this has a genotype 00, and this is a genotype 01, so a mutation here because it's the same person. This, ignoring those other quality metrics, which are quite the same. There's some technical stuff, but here's the genotype difference, right? This is the same person, and here the genotype 00 in his normal tissue, and in his tumor tissue we have 01. And I think we can see what 01 would be. Sometimes you have denoted AT, CT, GC, right? But in this case it's 01, and then you have a legend somewhere about which allele is 0 and which allele is 1. So the reference and alt fields contain nucleotide in case of SNPs and indels. In case of log checks for variances, those in certifications in versions. So this is the mandatory header line and the info. And here you can see the different alternatives: insertion, single nucleotide variable, deletion, SNP, deletion. This is the phase data, so GC and above are on the same chromosome, and this is reference allele GT is 0, right? Alternative allele GT is an index to the alternative column. So basically, you read this through this. And I guess you have this presentation and you can go into detail and try to figure it out for your own sake if you're interested. And some of you who's gonna do the bachelor's thesis with me, this is going to be very important. And we're gonna have obviously more talks about this in detail, but this is a good starting point for you guys to take it from there. I think it's, if I remember correctly, it's 3 of you. So this presentation is a good starting point, and I think you should also write a lot about those formats also in your thesis. And I think this is very good. You can take the pictures from here, and yeah. So this is the VCF 4.1, right? So this is how it looks. So genomic regions, subregion is defined by 3 required fields: the sequence header chromosome, start coordinate and end coordinate. Defined regions of interest are introns, exons, genes, etc. Extra information saved as field after the first 3 standard formats: BET, GFF, and GTF. They're tab separated with no header. So the mandatory fields which you need to have in the file are chromosome, the name of the chromosome, chromosome start, the starting position of the feature in the chromosome of scaffold 0 based, chromosome end, the ending position of the feature in the chromosome or scaffold. The chromosome end base is not included in the display of the feature. For example, the first 100 bases of a chromosome are defined as chromosome start 0, chrom end 100, and span the bases number 0 to 99. And you have the optional fields of name, score, and strand, which can display options like thick, start, and color blocks, the control, the view, and the genomic browser and such. And this is how it looks, right? Position starts, where it came from, the discrepancies. So the BET-P format, I haven't actually used this format very often, but nevertheless, this is how it looks. So saving BET-P format, 6 monetary fields, first region 3 fields, followed by second region 3 fields, followed by the rest of the optional fields. We have chromosome, the position, starting, ending, and then where it was sequenced, and is a positive or negative strand orientation. So BDP is a paired region, such as transposon cut and paste, a paired and read, a split clone, and inversion breakpoint, a translocation, etc. So this is BET tools. Again, I'm not a big fan of BET tools, but this is some stuff you can use. You can sort those BET files, merge, converge, overlap, subtract, and so on. Some similar which you used in Plink or any other software, right? And this is intersect, which I never used, but this is basically how it explains you how you put different genomic data merge into one. And this is also the same. So converting BUM to BET, you're given a BUM file with alignments for large scale reads. You want to extract some reads and save them in BET format to visualize them on a genome browser. And then you use BET format BUM to BET options, and then the input BUM, and you get out the stuff. So general features, your mandatory fields tab separated, so your sequencing name. The name of the sequence must be a chromosome or scaffold. Source, the program that generated this feature. Feature, the name of this type of feature. Start, the starting position of the feature in
Samo
this.
pitanje.
Yes.
Možeš vratiti slajd nazad.
Mhm.
Suštinski recimo, evo sad, da ne nađemo neke interesantne podatke i da smo ostali sa recimo whole genome sekvencama, mogli bi manuelno vaditi, ne znam,
Tako je.
gene koji nam trebaju za rad.
Apsolutno.
Okay.
Apsolutno. To je jedna stvar o kojoj sam razmišljao kako smo imali onaj sastanak. Zavisno od mogućnosti, da kad poskidaš jednu sekvencu, da izoliramo samo regije bitne za to što želimo pogledati, jel? Znaš, tako da ćemo razgovarati svakako o tome, ali odlično što ti je to isto naumpalo. Tako je, možeš jednostavno iskoristiti to mnogo lakše nego da imaš whole, ali sam ja mislio više kao da bi bio možda sam task od sebe, zanimljiviji da krenete kao ono, sa whole genome approach, ali ono, možemo tako krenuti kao da vježbamo i da na kraju kad budemo pisali rad da se fokusiramo samo na ovaj pristup u metodama. Ali o tom potom. Tu ćemo se dogovoriti. So general features, as I mentioned, in terms of start, score, basically, as you can see, all of this stuff is very commonsensical, right? There is no magic behind this. It's names and where something starts and where something ends, as a positive sense or anti-sense, to which group it belongs, chromosome position, etc. It's just the way how you use the text, the space in a text file, in order to write those things up, and how you use the 3D space of a piece of paper, so to say. And then you have here the GFF format. I've never used this, but it's very useful for molecular biology, for example, because they have all those information and on hand when they have a sequence which they maybe want to knock in or something like this, right? So the gene transfer format is an extension to a backward compiled GFF2. The first 8 GTF fields are the same as GFF. The feature field is the same as GF, with the exception that it also includes the following optional values: 5 UTR, 3 UTR, intra CNS, and intron CNS. The group field has been expanded into a list of attributes. Each attribute consists of a type of or value. Attributes must end in semicolon, semicolon, and be separated from any following attribute with exactly one space. The attribute limit must begin with 2 mandatory attributes. A global unique identifier for the genomic source of the sequence is gene ID value, and transcript ID value, a global unique identifier for the predicted transcript. So you can see why this format would be more useful for some molecular biology applications rather than genomics. And this is how it looks. So, but for me, not really. But if you get used to it, then it makes more sense. So sorting and indexing. Most tools require sorted input files. Sorting keeps things tidy, as we mentioned several times at this point. Raj files should be saved, sorted, and indexed. Indexing a file enables us to extract an element in instant time. All the file formats have index files, phi, sci, bi, and so on. So here you can see the FASTQ. We use some tools, phi index, sequence reads, SUMBUM, aligned reads, some tools, VCF tools for VCF, bed tools for bed, and so on. So, and this FASTQ file contains sequence of ATCs and Gs. Contains positions, chromosomes, starts, and ends, right? So you get from this, as we said, we want to FASTQ file to a VCF file where we have a string, letters, ATCs, and Gs, sorry, ATCs and Gs, and then you get a VCF file, which has all of this chromosome position, start, end, where all this was made sense out of with the steps, and this is what we got. Summary data formats, always use standard data formats. View edited data with standard tools. Cite the tools you use along with the version to make your work reusable. If you find a bug or need some utility, send a message to the authors of the tool or make an issue on their GitHub repository. Always check it if the tools are installed on SCC and load the module instead of installing it locally. That's true. Summary tools, when you want to work with these files formats, always think that other people have the same needs. There must be a standard way of doing it. Some tools, BAM tools, bed tools. So it's just go. This is what I said. Just Google this stuff, right? As soon as you have an issue, somebody actually solved this 20 years ago, and you just need to pick it up from somewhere. It's not that hard. So line reads to the reference sort and index, call SNPs, extract the SNPs from chromosome Y, overlapping all the elements. So it reads, see, this is kind of how you can read this file. I think this makes quite a lot of sense, right? It's like when English, like objective, predicate, subject, when you, what is what, right? You see, this is what we used all the time, except this bed tools part. You see, this is, we find what we want, And then we use alignment BWA, sorting the sum tools and GATK, extracts with sum tools U, and develop. Right? Super nice. So is our workflow, that data curating, and this is what we have tackled on the last time when we used it on the computer with the FastQ reads, FastQ reference, and have to check the quality of the FastQ, align reads, BWA, then you have sum with alignment, sum tools in index, which bum to alignment, remove duplicates, bum alignment, realign around the indels, GATK, bum alignment, and get statistics, sum tools, GATK. And then we have our genome browser. By the way, does anybody have any questions? I guess I'm going a little bit too fast with this presentation.
All good for me.
Do anybody else? Yeah, because I see I kind of rushed it a little bit too much. I went through 60 slides in half an hour. It's not really ideal, right? So let's have maybe a look a little bit here. So in our different, so hopefully at this point you have all installed the sum tools and the bum tools and all the stuff. Do I have bed tools? I don't have bed tools. We can use this time also to install bed tools. And you have it now recorded, so remember to install bed tools too. Good. That's great. See, all of the commands are well described in what they do. So you just need to call them up and have a look. That's VCF tools. That's what you also installed. BCF tools is also here. BWA. Let me see. Bow tie 2. I'll get you 2. Why not? Yep. See how nice and tidy it is. And this is extensions you can use. It's quite useful. Has anybody actually looked into the data since last time? Has anybody tried to analyze by itself or download some data? Anyone? No one? Okay. Can somebody remind me when is your final exam period?
22. januara mislim da počne ta sedmica.
22. do
Vjerojatno
29.
dvije sedmice.
2 sedmice? Stvarno?
Mislim da da.
2 sedmice. Dobro. Sad jedno pitanje, ali nemam vas dosta, ali ne veze, pa možete prenijeti. Da li biste voljeli opet da final bude kao koncipiran kao prošli put, što je midterm bio, ili biste voljeli da imate praktičan dio sa programiranjem, da pokušate kao da riješite neki mystery task koji je prejednostavan, ako pogledate prezentacije samo. Koji je vaš preference.
Prenići u grupu kad to neko odluči.
Ja, pišite vi ovako na prvu.
Pa slučno se samo ja tu, mislim.
Ma, može ko onaj prvi. I ja sam tu, samo šutim.
Dobro, da ćemo... Ja sam prepostavio da ću svakako vidjeti ko prvi, nego tam niko nije tu, pričam sam sebi, pa mogu kao da ispričam, znači da programiranje svima kontažno, niko ne odgovori ništa.
Ma jok, tu sam ja, ako nešto ja slušam pored Armine, ali nisam ništa radila.
Tu sam ja, nego sve jedno, većinski će to za prvi put, da se ispiše, to je to.
Slavno ti nas.
To će svi, ali nam duše sve jedno.
Dobro. Onda neka bude tako najlakše. Jer ja ću vam dati onda vjerovatno u ovoj sedmici ili idućoj sedmici pitanja. Zato bit će dosta obilnije nego imate vremena da mi dostavite do kraja prve sedmice ovih. Ako 22. počinje, onda 29. znači mjeseci, 5 sedmica ćete imati, na isti fazon će biti, ali će biti dosta obilnije. Znači manje više ćete ispisati, neće reći ko diplomski rad, ali koja baš dobar esej. Baš dosta. Zato imate toliko vremena da svaki dan pomalo možete uzeti da radite. I nadam da ćemo tako nešto riješiti. Igor, Irnes i Armin, jeste razmišljali među vremenu nešto malo oko ovih teza, šta, kako, gdje?
Ja iskreno nisam, pošto smo imali nekih prezentacija, puno iz dosta predmeta, tako da da to malo samo smirim, pa ću onda da se bacim na te teme.
Dobro,
znači, što se tiče mene, zadnjih 20 minuta.
A što se mene tiče, ja sam definitivno vratio nešto, mislim, volio vratiti sa nekim pitanjama koje se vežu za neki rak, ali ne znam, nisam ništa konkretno tražio, ono što smo spomenuli prošli put, da su što kompaktnije sekvence i da je lakše raditi s njima, ali da su vezane za rake. Sad koliko je to izvodljivo ne znam, ali to je nešto okvirno, ali nisam ništa konkretno.
Ja sam isto za opciju da nije čitav genom, pošto kako zove, imam 8 giga rama i sad je to procedura, morao bi baš para da dajem da bi čitav kompjuter mijenjao, pošto mi je matična, da sad uglavnom ne radi mi se to iskreno. Mislim, ako je neophodno, uradit ću, ali...
Znate kako naravno da nije neophodno, ne mogu da se natjerati trošite pare, mislim, to vam pameti. Nego smo pričali o tome, vi da, ako hoćete i da radite segmentirano ovo, sjećate se, morate prvo skin svakako sve. Morate svakako raditi alignment prvo i onda nakon alignmenta izvaditi te dijelove ponaosob. Tako da najduži dio koji je taj alignment koji će vam trajati preko noći za jedan file. Zbog toga ja primarno mislim da je to, jer kasnije analiza, kad se ti failovi kompresuju, kad se uradi training i sve ostalo, ovih višaka, nazovi tako, ti failovi postane relativno kompaktni od nekoliko stotina megabajta, eventualno gigabajt 2. Nego ovaj najveći, najkrupniji dio posla je zapravo sam taj početak da bi uopšte mogli da katamo, naravno ćemo smanjiti dijelu analize i dosta vremena, jednom kad znamo što treba da analiziramo, onda i to iziskujemo drugačiji pristup, gdje ćete vi istražiti sami to i meni reći gdje su te regije koje treba da pogledate i zašto želite da pogledate regije, ako me razumijete. Recimo jedan prijedlog, pa mogu i drugi uzeti to u razmatranje za svoje radove. Kaže, pošto redamo public available data, ljudi koji vam reći nisu bolesni, nego su onako random sekvencirani, nisu to kancer uzorci. I kažemo, znamo da je predispozicija, čuli ste Angelina Jolie, što je čuveno bespotrebno osjekla sebi dojke i izvadila jajnike, ima ta BRCA1 mutacija, koja ima podešava predispoziciju ginekoloških oboljenja i onda kao preventivno uradiš hysterectomy, kako se kaže na našem, i riješiš se svega toga i sad recimo možete vidjeti koliko ljudi u generalnoj populaciji nosi tu mutaciju za sobom, a kao just a random screen up on the population level, what's the allele frequency of that allele na random population I choose. I znate gdje se nalazi BRCA1 i samo izvadite taj kromosom i samo gledate tu, jel? Ali da biste došli do toga da možete izvaditi taj jedan kromosom, morate prvo raditi alignment. Alignment su na 2 FASTQ fajla plus reference file, znači 6-10 GB najmanje u svakom trenutku treba vam za to. I onda, jesi tebe, Armine, 8 GB RAM kompjuter, je li tako?
Nije imao više.
Imam 16, da.
I čitavu noć ti je vrtilo, jel?
Ma nije, mislim da je jedno 4-5 sati je trebalo, a bio je file 25 giga.
25 GB. Znači, eto računaj da tebi onda ne se treba preko noći, da ostaviš to pod uslovom da si sve setupovo super i da upravo frke ne prekine kontaž, radi 2 sata i nešto si pogrešno ukucao, nisi vidio, da džaba vrtila, razumiješ?
A ja, pa već sam ja to jednom izvrtio, jednom je preko noći to završio sam.
Tako da otprilike morate planirati dosta vremena za to. Mislim, kažem vam, jednom kad se skontate da to funkcioniše, onda vam samo trebate pustiti to navečer da radi i otići spavati, razumiješ? I ujutru izvući podatke i kasnije analizirati. Tako da, razmislite, ja ne znam imate li vi pristupa na burču tu nekom kompjuter roomu ili nešto, postoji li to uopšte tu?
Imaju na burču full labovi sa kompjuterima koji mislim da su sasvim ok za ovo, tako da...
Pa ne znam, imate pravo da koristite to, imate li da?
Nastaje problem na burči, pošto im struje nestaje dosta, i zna se desiti.
Je da radiš nešto da te prekine, jel, nestanak struje?
Nema problema.
I friždera i svega, ne znam, nije baš stabilno što se toga tiče.
Ne, ako to baš bude, kako zove, vidjet ću, ako bude, završit ću ja to ispirati.
Nema, niđe veze baš zbog bachelora da daješ sad 2-3 glave rama matičnu kontaž, mislim, niđe veze kontaž.
To mi je samo, ne mogu 2-3 glave, morao bi 1000 dati. Jer kako zove, morao bi matičnu čitavu da mijenjam, onda 16 GB rama DDR4, onda procesor mora mijenjati, jer ovaj procesor ne bi mogu na novu matičnu, morao baš sve da mijenjam, zato.
Ma lakše kup novi kompjuter nek to zabacati.
To.
Nego nema mi smisla da je doktorat ili nešto pa da kaj, hajde to neko investicija.
Da je to nešto aman zaman, kako zove, nego diplomski sad da mijenjam čitav kompjuter, baš mi to nonsens.
Pa niđe veze, ali zato treba da to na burč, majke, ima plaća.
Ma ja, skontat ćemo nešto.
Otičite, tu neki taj kompjuter lab, sjedite, napravite pipeline i pusti nek vrti, razumije. Ako ništa iskoriste priliku da poskida, ili im brz internet, nije, ni to, jel da? Znaš,
Nope.
skontali ste da se to skida relativno dosta, razumiješ, evo, znači kad skidate otići negdje imate konekciju, kako Bog zapovjeda, da se ne treba 12 sati da skinješ fajl, razumiješ.
Ja koliko znam što se tiče tih labova sa kompjuterima, da su oni generalno u vlasništvu odsjeka za one grafički dizajn. Ja ne znam koliko su oni lenient da daju nama akses na njihovim kompjuterima.
Imaš pristup uvijek tim učionicama, to bez problema. Može se koristiti jedini problem internet.
Aferno.
Sad mislim, ako neko ima hard disk, može skine kod kuće, ne znam, 100 giga pa prenese vamo, prebaci.
Ja, to je isto super kontaž, to pametan pojam. Nego šta znam, jer računajte koliko vam vremenski treba da poskidate to. Znači, ako ćeš uraditi, ako ćemo 20, ajmo reći 10 kontrola, 10 case-eva koji ćemo randomly assign, pa 2 FASTQ fajla. Znači, ako ti treba da svaki FASTQ file 2-3 sata, ko što vam je trebalo, ja mislim, i više čak, znači, to ti je 60 sati skidanja samo. Sataš, bez da si se počeo raditi, znači nisi ništa uradio. Već 3 dana pravio skidajući te stvari, tako da mislim da morate malo to ispredati. Ja vam rado pomogao kako mogu oko te logistike. Ja ću dolaziti sad opet 24. februara, bit ću 7 dana tu, neku dokumentaciju završavam, ponijeću kompjuter i sve ostalo, pa ako bude negdje zapeo da u tih par dana gledamo da odradimo nešto zajedno, da analiziramo dok sam ja tu, na mom kompjuteru, pa onda ću, da ja poskidam kod sebe kući, pošto imam kući drama brz internet u koloniji, pa onda možemo da vidimo i tako. Uglavnom, da riješimo to što prije, da počnete pisati, da počnete raditi odmah poslije nove godine, da to završimo, znaš, da ne dužimo i zbog vas i zbog mene, da možete dalje se upisivati za master i šta ti ja znam.
Važi, može.
Eto, i sad ćemo samo još da vidim koliko sam ja slajdova ovdje došao. Ja, ovo samo na brzini da vam, znači ovo bukvalno, ovo je super ova prezentacija što vam je poslao, je tačno opisuje ono što smo govorili i radili i u principu nema tu previše vrdanje izvan ove logike. To je ta logika, vaši diplomski i ovo što sam radio nije 10 godina, nije 10, 7, je ovdje sažeta u ovom veoma dobro. I mislim da ova prezentacija baš objašnjava sve te pikanterije male koje su između i plus ono što imamo znanja iz onoga što smo radili ranije. Ako ništa logički objašnjava što se događa. Jer lagano je naučiti sintaksu pisanja nekog koda ili neke komande. Ova komanda je ovo, ova komanda je ono, na kraju krajeva ne trebaš naučiti na pamet, možeš uvijek imati neki cheat sheet kraj sebe i zanima me ta komanda, to ću ukucati da bi dobio ovo. Nego šta je mnogo bitnije ovdje i zašto je ovaj kurs po meni treba da bude ciljan i da ta logika, kako od ove tačke ovdje ovo što ja dobijem doći do ovdje. I to je u principu ovo sve između što mi govorimo. Ima razlog zašto ovo ide sa nekim redoslijedom. Ne možemo bez alignmenta uraditi sortiranje. Bez sortiranja ne možemo skloniti duplikate, jer da ne znam šta je duplikat. Bez sklonjenih duplikata ne možemo da realignamo oko indela. Bez toga ne možemo da uradimo variant calling i tako dalje. I kad shvatimo, mislim, to kao konceptualno, onda je ovo jako jednostavno zapravo, mislim. Kažem jako jednostavno, naravno da je jako jednostavno za vas u odnosu na vaše intelektualne sposobnosti, dovoljno ste inteligentni da se bavite tim što bavite, da ste došli 4 godine određenog studija koji zahtijeva neko mozganje s glavom, koja s te strane kažem nije teško, jel? Naravno da nekome ko radi negdje nekoj drugoj poziciji u životu ili nije razvio svoje sposobnosti u tom pravcu, ovo predstavlja ogroman teret i ogroman problem, ali mislim da vi imate svi mogućnosti da ovo savladate bez problema, jer se ne bi nalazili tu kroz polaganje raznih drugih ispita tu gdje ste sada. Tako da ovo ne treba da bude ništa strašno i da moj cilj jednim dijelom je potpuna demistifikacija ovih rabota, tako reći, koji se često predstavljaju kao neka naučna fantastika bespotrebno. Mislim da je to za svaku profesiju važi, da ta bespotrebna mistifikacija odvraća mlade, kako da vas nazovem, od ulaska u ova polja koja djeluju visoko sofisticirana, ali u principu jednom kad čovjek savlada rezonovanje iza ovog svega postaje jako jednostavno i streamlined. Tako da, opet ponavljam, jedan od glavnih razloga je ako ja ovakav mogu, to možete i vi sigurno. Tako da, ja ovo stvarno mislim, nije neka šuplja motivacijalna priča, nego jednostavno, mnogo sam ja bio limitiran i vaših godina nek što ste vi, tako da bez problema vi ovo možete savladati. Provedete svaki dan po pola sata i kucali biste ove pipeline za 6 mjeseci. Dobro, uz to sve imamo ovaj Genome browser, njega ste vjerojatno koristili na ostalim predmetima koji se zovu bioinformatika u BiH, na svim fakultetima na kojim sam ja do sad imao dodira s ljudima, gdje bioinformatika predstavlja ovo, bukvalno ovaj Genome browser, BLAST i ovih par opcija što se nalazi ovdje, i da oni kao to baš prolaze studentima i to je bioinformatika i tako sam ja završio bioinformatiku. I u principu, zašto vam ti toolovi služe? Jednom kada imate neki find koji ste vidjeli da je signifikantan u odnosu na neku normalu, kad ste uporedili te svoje sekvencirane i dorađene fajlove, onda ukucajte tu genetsku regiju ovdje u ovaj browser ili neki sličan browser i vidite šta ostale studije i generalna literatura kaže zašto je ta regija bitna i šta se može sa njom raditi, koliki je CG content, da li uopšte možeš napraviti primer, kakav bi bio, da li možeš uraditi RNA interference za ekspresiju. Sve te ova stanica kaže ima more ovih subbaza koje su svaka podijeljena na neki drugu sferu interesa koja ti govori za tu genetsku regiju i poziciju šta je sve moguće. Tako da je ovo jako dobar tool, ne kao samo za sebe da vi nešto tu istražujete i radite, to je niđe veze, nego kad ste već nešto istražili, uradili, analizirali, ovdje možete da verifikujete to svoje razmišljanje, da li je već možda neko pogledao to. S druge strane, recimo imate fenotip koji je lupan, da ne znam, neko mi se grči noga, ne znam, to ste vidjeli kao, to je neka čudna mutacija koju bacite ovdje, vidite da li je ta promjena u blizini nekog gena šta ja znam, koji radi nešto sa neurotransmisijom, miostatinom, nekim tako, guess u kojem pravcu da idem. I zato je ovo super cool tool. UCSC liftover, so each reference assembly version has a slight improvements, gaps that were closed, repeats that were expanded or contracted, errors that were corrected. A coordinate in one assembly like HC90 will not always correspond to the other HC38. We will need to convert between them. That's called so-called liftover. To convert corners between one assembly to another, use the liftover tool. So should you use build 38 to build 37, I usually still use 37, but a lot of people are now switching to 38, even though 38 is 10, 15 years old, but 37 seems to be more stable in analysis. That's just some of my experience. Choosing the reference of the latest version always is the most precise, kind of, but it takes a while until the annotations, G nexons, repeats, binding sites are found and reported. That's what I was thinking. If you use the latest version, you will have better alignment, hopefully, but post-processing the functional biology would be difficult, and that's why we stick to the build 37. I think we will stick also for your bachelor's thesis to build 37 rather than 38. Except if there is any preference in the university, but I don't think why they would have a preference in any of those. If you're studying genes or functional features, always use the second latest version. Yeah, this is build 37. So this is the liftover. How you do it? You put this file here, put the sequence here, and then it changes the sequence accordingly. But I think also, let me see if there's a liftover tool here. It's an executable like Plink. Free. Let me see if it works, because it's only for Linux, right? But let me try to see if we can call it up. Yes, it works. Good, so this is something you can use to interchangeably change your build for one to the other. Sometimes it's useful, sometimes, but I would just stick to build 37. It's much easier, I think. With that said, do you have any questions? Anything about the presentation or anything further which might be interesting to the people who are listening? No? Okay, cool. I think that's it then for today. Next week we'll, and I have to get this stuff going to check on while the file line 94 was not working, but I'm postponing and procrastinating on this since I came home on Sunday. So we will, we will most definitely continue with programming the next two meetings, and I have to force myself to check up on the file to see what's going on. I would be really pleased if someone could send me a message and remind me that I should do this. One of you guys next week or this weekend, just write me a message, do it. Maybe one of my thesis students can just remind me, just do the line, please. The line of code, I mean. Yeah. Good, guys. Thank you so much for listening to me, like always. I hope something is more clear, but you can also go through the presentation yourself. It's very self-explanatory, I assume, and then we take it up then next day or next week when we see each other. Okay, good. Bye-bye.
Well,
Bye.